for your gene of interest. And that's pretty much the way it goes. Now with northern blot, you don't really have to do the cDNA step unless you're going to be doing reverse transcription PCR, where you don't have enough starting material for northern blot to be able to actually uh, visualize the probe binding to the mRNA. So, you know, northern blot, you're comparing the intensity of the band. And if you have a band that is very, very high intensity and is like totally blown out versus another band that is very thin and is not very high intensity, then you can, do like a qualitative comparison saying that, oh, well, the larger band means greater expression of this particular protein of interest. And then you remember we use a housekeeping gene as a control to make sure that the reason why we're seeing this huge band is not because we actually got more mRNA, but is because it is actually being overexpressed in maybe the cancer liver tissue versus the normal liver tissue. So when we do northern, it's more of like qualitative, like you're looking to see the intensity of the bands. But when we're doing qPCR, it's actually called quantitative PCR because now we we get a higher level of resolution. We can actually count the number of mRNAs that we started off with. We can actually determine how many copies of mRNA were present. And we can also use qPCR to determine, just expression levels, but we can do it quantitatively as opposed to qualitative. Like, oh, this band is, more intense than this band. That would be like your northern blot, where quantitative PCR, you can actually be counting the number of mRNAs, and that's what makes it so powerful. So this is like a more updated approach to northern blot. Northern blot, um, the materials that you need for it are 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 not very expensive at all. Okay, I mean, we talked about it. The materials you need for northern blot are not very expensive. For real-time PCR, it is more expensive. You have to have a special thermal cycler that's different from the one that you're going to do normal PCR in. It's got to have, each well has got to be able to go and get hit by a laser, and not only that, but you got to have a sensor to measure fluorescence in each of the wells of that PCR unit, of that thermal cycler. Okay, and so what we are going to learn with regards to qPCR is that there's two ways that we can do it. One way is by using something known as cyber green, which is just basically it's a DNA stain, and it preferentially stains double-stranded DNA versus single-stranded, and you can buy it anywhere. Okay, and it's called cyber green. Okay, the other way that you can go is by using something called TACman probes. And TACman probes are proprietary. So, if you're going to buy TACman probes, you got to go and buy it from Thermo Fisher, which is the company that makes these probes. These probes are very sophisticated because on one end of the probe, you're going to have a reporter, which is on the 5' end. On the 3' end, you're going to have a quencher. Now, the reporter is basically a fluor that when you hit it with, laser light, that it will fluoresce. But if the fluor and the quencher are intact, meaning that they haven't been separated from each other, you won't get any fluorescence. And so we'll see how this works on the next slide. But there you go. Okay, so I don't want to go any more into the slide because we've been through it. So let's go and start learning about these two different ways that you can go and do qPCR. So over here on the top, this is going to be our TACman probe. Okay, so TACman probe, as you can see, it's a single-stranded DNA probe, and it's not very long. Maybe it's like 20, 25 nucleotides in length. It's not a very, long stretch of DNA. And on one end, the 5' end, represented by the green ball, you have your reporter, and on the 3' end, you have your quencher. And when the reporter and the quencher are intact where they are, you can go and hit the reporter with laser light or UV light, and you're not going to get fluorescence. Okay? And so you might wonder, well, how does this work? Well, the thing is that when you do these experiments, like let's pretend that you're interested in a particular protein. And you remember we said you maybe believe this protein is, produced by way of an oncogene, a gene that causes cancer. And you want to know if in cancer liver tissue versus healthy liver tissue, you want to know if there's a higher expression or higher level of expression of that oncogene within that cancer liver tissue. So what you would do is you would design the probe to be able to bind to that that you don't want to say mRNA, because when you go and you do TACMAN, you're taking your mRNA and you're converting it into cDNA. So your cDNA is a copy of all the mRNAs that were there, right? And so what you're going to do is you're going to go and design your probe to plop down somewhere within your gene, within the cDNA of your gene. Okay, and over here we could see that it actually happened, right? So when you do qPCR, you are doing PCR, but with your PCR reagents, you have these TACMAN probes. Okay, and so when you go and you do your denaturation step and your cDNA denatures, your TACMAN probe is going to bind to one of the strands, as we see right over here. And look, here's a primer. So what does your Taq polymerase do? It's going to go and latch on where the primer is, and it's going to go and extend or synthesize in a 5' to 3' direction. And when the Taq polymerase comes across this TACMAN probe, right? Because so you're going to go and denature. You're then going to go, and your denaturing temperature, we said is like somewhere around like 94. Then you have your annealing temperature, and the annealing temperature is something that you can calculate. But usually it's between like 52 and 58, and that's when your TACMAN probe will bind to one of the strands. And then you have your extension, which is around like 72, 74 degrees, and that's when your TACMAN probe will start to go and synthesize in a 5' to 3' direction. So what happens when it goes and encounters the probe? Well, it turns out that it has 5', 3' exonuclease activity. That's what Taq polymerase has. So if there's anything that is already attached to that single-stranded DNA, when it comes to it, like the probe, it just goes and just eats it up. It just goes and destroys it and plows right through it. And so what happens when the Taq encounters the TACMAN probe? It will end up releasing the reporter from the quencher, and then when that little well in your thermocycler gets hit with a laser, the reporter then has the ability to fluoresce, and that will get picked up by a sensor in the well of that thermocycler. Okay, and that's how it works. So what it is, when you go and you do TACMAN probes, you have a huge number of TACMAN probes. Maybe you have millions of TACMAN probes in that small little PCR tube. Okay, but when you hit it with laser light, you're not going to get any fluorescence until the probe is able to bind to double-stranded DNA, and the Taq ends up plowing over it. So this fluorescence that we see from the reporter, that fluorescence that you see is basically a proxy for how much PCR product that you have. Okay, that's what it is. So it's basically a proxy for the amount of PCR you have. So if you only have, two double-stranded DNAs, and then you do PCR, okay, then when you denature, the double strands separate. And then when you anneal, you would have, there's only a place for two TACMAN probes to go, one over here and one over here. And then when the TAC polymerase ends up going and synthesizing 5' to 3', those two TACMAN probes will have the reporter released from the quencher, and now you have two reporters. The next round, you would have double the number of reporters. The next round, you will have, once again, double the number of reporters. So the amount of fluorescence is a direct correlation with the amount of PCR product. Okay, and that's what makes it quantitative. Now over here, what you have is cyber green, and cyber green is similar, but I want to see if you can tell me what is the drawback. What is the drawback for using cyber green? But first, let me explain. So the whole idea with cyber green is that it's this stain, a DNA binding stain that preferentially binds double-stranded DNA. So as you accumulate PCR product in the form of double-stranded DNA, you have more of the stain that is able to bind. And so once again, the amount of fluorescence in the form of cyber green is a direct correlation with the amount of double-stranded PCR product that you have. Okay. Now it turns out cyber green is like pretty freaking cheap. Okay. Cyber green doesn't cost a lot. TACMAN probes, they cost a lot because when you go and you do TACMAN probes, you are basically telling them to synthesize a probe for your gene of interest. And they got to actually make it. Where cyber green, it just goes and hits any double-stranded DNA. Okay, so based on this information, why do you think TACMAN probes would be preferable versus cyber green? Who can write that down in chat? Why would TACMAN probes be preferable to cyber green, even though cyber green costs a lot less? It costs a lot less to go and do qPCR with cyber green. Okay, let me go and stop sharing to see what you guys are putting in the chat. Yeah, absolutely. Absolutely. You guys are great. Okay, that's right. It all has to do with specificity. It all has to do with specificity. So let's go back over here and take a look. You guys are great. Okay, so over here, cyber green is going to bind to any double-stranded DNA molecule. So if you're doing your qPCR and maybe you're amping up something else besides your gene of interest, maybe you have mispriming, maybe there can be 110 different things, but that cyber green is not specific. It just will target any double-stranded DNA. Where on the other hand, even if you do have some degree of mispriming, or your primers are binding somewhere else to maybe another cDNA, it doesn't matter. And the reason why it doesn't matter is because your probe is specific for your particular gene of interest. And that's what makes qPCR, or I should say that is what makes TACMAN probes, like kind of like the gold standard for qPCR. But if on the other hand, you are totally sure that the amplification you're getting is strictly your gene of interest and you're not amplifying anything else inadvertently, then cyber green can work. And it depends on, are you doing preliminary, like a preliminary pilot study, or is this something that's going to be published? And if it's something that's going to be published, then maybe you'd want to go with TACMAN probes, depending on what your question is. Okay, so there you go. So let's go and move on to the next slide. So now what we're going to do is we see in text what I already explained to you guys verbally. So we got two approaches. When we go and we do qPCR, we always start with an RNA extraction, and we're going to use reverse transcriptase to take all of that RNA from that tissue sample. There can be hundreds and hundreds of different RNAs, mRNAs. And what we're going to do is convert it into cDNA, because you can't do PCR on RNA. You can only do PCR on double-stranded DNA, and that's why we need to get our cDNA. So TACMAN probes are complementary to specific regions of our target cDNA, a particular gene that we're looking at. And the cool thing about TACMAN probes is that, I mean, if you set it up right, you could probe for more than one gene at a time. You could do that. And so you would just make sure when you order your TACMAN probes that the different probe, when the Taq polymerase plows through the probe, releasing the reporter from the quencher. Now, Cyber Green is not as specific. It just is going to bind double-stranded DNA, preferentially over single-stranded DNA. And the more double-stranded DNA you have, the more fluorescence that you get. And fluorescence is going to be directly correlated with the amount of PCR product, and the same thing with the TACMAN probes. Okay, so this is really good. So what this shows right over here is how the output would look like. What would the data look like when you go and you do qPCR in a qPCR thermal cycler? Okay, what is it going to look like? And over here, what we see is in A, we've got our double-stranded cDNA, and we're going to do PCR. So we got to denature the strands. We're going to go and go back to an annealing temperature. And at our annealing temperature, this is where our forward and reverse primer are going to bind. And right over here, your quencher is going to bind too. And when you do your elongation step or your extension step, your Taq polymerase is going to plow through the TACMAN probe, releasing the reporter from the quencher. Okay, and so this shows what is happening in the tube. Over here, this is they show your Taq polymerase as like a Pac-Man. And you know how Pac-Man goes like this, the video game, and it just goes and gobbles up the nucleotides, making up the probe, releasing the reporter from the quencher, and then you're gonna get fluorescence. Okay, so this is what the output would look like. And on the x-axis, okay, which is going to be your dependent variable, you have your number of cycles, number of PCR cycles. So this is your, 0 cycles. This is at 4 cycles, 8, 12, 16. So obviously, after 0, you would have 1, 2, and 3, and then 4, and then 5, 6, 7, then 8, and so forth. Now, this right over here, your independent variable on the y-axis is the amount of fluorescence, okay? This is the amount of fluorescence that is getting picked up in the individual tubes. So what we're looking at here, for example, is just what's going on in one tube, in one single tube. And what we see over here is that as we continue through our PCR, that we see that as we continue through the PCR, that we have what's known as a threshold cycle. Okay, we have a threshold cycle. So what we could do is we can do this. Now, to really understand how this is working, what I would say is I'm going to go and correct myself. Okay, so what we're looking at right here. No, I'm not going to correct myself. Okay, I'm legit. I'm going to continue with my explanation. Okay, so this is happening. Hygiene expression. No, I am going to correct myself. I'm going to say what we're looking at here are actually in different tubes, and each of these lines that we're looking at, okay, each of these lines, they're representative of a different tube, okay? of a different tube in the thermocycler. I think this is going to help you to understand how we go and read the output. Okay, so right over here we got two different tubes, and each line corresponds to a different tube. And what we have in red is the threshold cycle. So what the threshold cycle means is that any fluorescence that is below the threshold cycle, we're going to say that's background noise, that it's too faint to really be considered much of anything. Okay, so below the red line is, it's the fluorescence is too low to kind of even, uh, make anything out of it. So we're just going to say that below the red line is like background noise. But once you pass that red line that we call threshold cycle, then we say, okay, we've got enough fluorescence that what we're seeing is legit, that it's not just background noise or fluorescence. Now you might be saying, well, what does this mean? What does it mean that, for the qPCR in this tube versus the qPCR in this tube? What does it mean that this line right here past the threshold before this line right here? What it tells us is that right over here, this line that says high gene expression, why did the line or a curve for high gene expression, why did it cross the threshold cycle before the line that we're going to label as a low gene expression? Why did the high gene expression cross before the low gene expression? And it all has to do with how much initial mRNA you started out with. So if you're looking at a particular gene, and if you start out with 1,000 mRNA copies of a particular gene that you're looking at, that you're researching, if you start out with 1,000 mRNA copies, and that is converted into cDNA, now you have 1,000 cDNA copies. Then when you start your qPCR going one cycle, you're going to double from 1,000 cDNAs to 2,000. And then the next cycle, 4,000, then 8,000, then 16,000. Okay? And that's a lot of fluorescence. On the other hand, for the tube that has low gene expression, what that tells us is that the amount of starting mRNAs for our gene of interest was dramatically lower. Maybe instead of having 1,000 mRNAs, like we saw for high gene expression, maybe there's only 50 mRNAs. So after the first cycle, you only have 100 PCR product. And then after the second, you would have 200, then 400. So now you can see why you would pass the threshold cycle for fluorescence if you have more mRNA transcripts. The more mRNA transcripts you have, the faster you're going to reach that threshold cycle. And that's how we do qPCR. Okay? So if, for example, these two curves represent two biopsies, maybe one of liver cancer biopsy, and the second tube would be your mRNA from normal healthy liver tissue. And once you do the mRNA extraction, and each, that's present in with each of those tubes, and then you convert to cDNA, the tube that is representing the cancer liver tissue is going to have maybe more expression of that oncogene versus the normal liver tissue that would have very low expression of that oncogene. And so the lower expression, the fewer mRNAs you have, the longer it's going to take to build up the fluorescence in your qPCR reaction to pass the threshold cycle. Okay, so that's how it works. That's how it works. And then you might be saying, okay, but so we can look at the threshold cycle to determine where we had more mRNA, okay, where we had a lot, we can determine where we have higher levels of gene expression based on when they cross the threshold cycle. And the important thing to know, it's not really a factor of how high the curve goes. The big factor is when you cross the threshold. If you cross the threshold first, that's going to be high level of gene expression. Versus if you cross the threshold later on, it's low. Like right here, we crossed the threshold at the 12th qPCR cycle. Over here, you crossed it at the 20th. Why did it take so much longer? Because you had far fewer mRNAs with the sample that had low gene expression. And then what you could do is that you can use qPCR to actually figure out the number of mRNAs by using um, controls, by using a known quantity of starting material, by using a known quantity of cDNA. So, for example, you could go and do positive controls to get like a standard curve, where if you know this tube has 100 cDNAs and this tube has 200, and then this tube has 500 and this tube has 1,000, then you can see for those different tubes when they cross the threshold cycle. And then what you can do is you can go and graph that, um, and then you can figure out what these unknowns would be. Okay, so if you if you have known starting material, then you can do a series of dilutions, and then you can run that on the qPCR, and then you can generate a standard curve because you know how much starting material you had as to when they're going to cross the threshold cycle for fluorescence. And then you can do a little plot, and then with that information, you could take your unknown data and you can plot when they cross the threshold cycle, um, and figure out how many mRNAs or cDNAs did you start off with. And that's why it's qPCR. Okay, so this is super cool stuff. If you didn't watch that video, which most of you did not, go and watch that video. Okay, super cool video. Um, let's see how we're doing on time. Okay, so make sure you do that. Actually, we're gonna watch the video now fast. Okay, we're gonna watch it now because I know that you guys, some of you are not gonna be watching that video. So I want you to be able to see this video. It's pretty cool. There are two videos. One is how TACMan probes work. You're gonna do that on your own time. But over here, analyzing qPCR, that we can do together. Okay, so let's go and do that. Okay, and I'm gonna go and share my screen so we could see. Okay, let me just make sure it's opening up here. Okay. Okay. Okay, there we go. So what I'm gonna do is I'm gonna go and share. Now, the one thing I'll tell you is that the narrator, she speaks like crazy fast, and she has an English accent. So it might be hard to understand. But if you watch this later on your own time, you can go and slow down the video so that way she talks at a normal rate that you can understand. But let's take a look, okay? Let me optimize for sound and for video.
qPCR, a quantitative real-time PCR, is simply classic PCR monitored using fluorescent dyes or probes. qPCR is accurate, reliable, and extremely sensitive. It can even detect a single copy of a specific transcript. qPCR is commonly coupled to reverse transcription to measure gene expression. No wonder it is so important for molecular diagnostics, life sciences, agriculture, and medicine. Firstly, let's go over the nuts and bolts of qPCR. For this you use a fluorescent dye which binds the DNA. As qPCR progresses, the fluorescent signal increases. Ideally, the signal should double with every cycle, which is then plotted. Because there are few template strands to start with, initially there's a faint signal. Eventually, usually after 15 cycles, the signal rises above the background noise and can be detected. This is the threshold cycle, CT, the point from which all quantitative data analysis begins. But how do you analyse qPCR data? You can either use absolute quantification using a standard curve to quantify a sample from one treatment, or relative quantification using one or more reference genes to standardise and compare the CT values between two treatments. The absolute standard curve method determines regional DNA concentration by comparing the CT value of the sample of interest with the standard curve. To create the standard curve, you need to make DNA samples of known concentrations. After doing qPCR on these, you will see different PCR plots for each standard, and unsurprisingly, they have different CT values. The greater the concentration of the original DNA sample, the smaller the CT value. So if you plot original DNA concentration against the CT values, you have a standard curve like this. Now let's say the PCR plot of your unknown DNA sample is somewhere here, which corresponds to this CT value on the standard curve here. Using the standard curve, you can find out the log concentration of your DNA sample to be X. As this is in log scale, you can simply calculate your sample DNA concentration to be 10 to the power of X.
So this right here is really powerful because you can actually figure out how many mRNAs you started out with for a particular gene of interest. And that's what makes it quantitative. It's not this band is brighter than that band, but if you go and you do the standard curve, okay, you can actually figure out how much, the level of transcription that has occurred. And that's something that's really powerful.
Absolute analysis is suitable when you want to determine the actual transcript copy number, that is, the level of gene expression. On the other hand, relative quantification is used when you want to compare the difference in gene expression between two treatments, for example, light or dark treated Arabidopsis individuals. This is done using one or more reference genes, such as actin, which are expressed at the same level for both treatments. You then perform qPCR on both your samples and the reference gene. Find out the difference between the two CT values, delta CT, in each treatment. Now the ratio of the two delta CTs tells you how much gene expression has changed. For instance, in the dark treatment, the CT value of your reference gene is at this level, the CT value of your target gene is at this level, so you have this delta CT, which is the difference in CTs in the first treatment. In the dark treatment, the CT value of your reference gene is still at this level, but the CT value of your target gene may become only this much. So the ratio of the two CT values is delta CT for the dark treatment divided by delta CT for the light treatment, which equals 1/3, showing that delta CT has decreased by a factor of 3, which means that gene expression of the target gene is greater in the dark treated sample. This is how relative quantification using a reference gene helps detect change in the expression of your target gene. In conclusion, there are two ways to quantify transcripts using qPCR: absolute quantification using a standard curve and relative quantification using a reference gene. The method used depends on whether you want to determine the actual number of transcripts or the relative change in gene expression.
Okay, there we go. Okay, raise your hand if that helped out with regards to a better understanding of qPCR. Okay, she does. I told you, she has a strong accent and she does talk very fast. But if you go and you watch the video on your own and you're able to slow down that video, then you can understand everything perfectly. Okay, so let's go back and continue. Let me go and get this modified for, okay, for PowerPoint. Okay, and let me go and move on. Okay, so now what we're going to do is learn about fish. Not the stuff that swims in the water, but fluorescence in situ hybridization. So in situ, you probably know about in vitro and in vivo. So in vitro means in glass, like in a test tube. In vivo means within the organism. And in situ means on site. So what are we doing with fish, fluorescence in situ hybridization? We can use fish for a lot of different things. It is just another tool in the toolbox of somebody that is utilizing biotechnology. Okay, so we have a lot of tools now. We've learned everything from how to utilize restriction enzymes, how to do a DNA extraction, how to do reverse transcriptase, how to go and use northern blot, southern blot, qPCR, and now we have fish. So we use fish to basically determine chromosome location of a particular gene and also copy number of a particular gene. So how does this procedure work? Well, with regards to karyotyping, um, and so we do karyotyping to make sure that maybe an embryo um, does not have a particular genetic ailment, like a horrible genetic disease. That could be one reason why we're doing a karyotype. We can also use it for normal diagnostics. There are different cancers that result because of a translocation of genetic material from one gene to another. And as a result, the cell becomes cancerous. And so we can use this as a diagnostic procedure, which is pretty powerful. So this is how it works, okay? So chromosomes are isolated from cells, and you're going to spread them out on a glass microscope slide. At that point, what you're going to want to do is denature the double-stranded DNA, because that's how our probe, which is single-stranded, a single-stranded oligo, is going to be able to go and bind to that target DNA. So you can use either a DNA or RNA probe for a gene of interest that is labeled with a fluorescent dye, and you're going to incubate it with the slides. The probe is then going to hybridize with its complementary sequence with that target area on whatever chromosome that it's meant to hit. You're going to go and wash the slide in order to go and get rid of any unincorporated probe. And then you're going to go and expose the slide to fluorescent light using like fluorescent microscopy. And wherever the probe is bound to the chromosome, you're going to get fluorescence, and you're going to be able to go and pick that up. So do karyotype to determine which chromosome shows fluorescence. And this over here is in our textbook. This shows that we have our probe DNA, and we're going to go and label this probe with a fluorescent dye. And so that way we can then denature this double-stranded probe. So then it's going to go and complementary bind to its target location. So fluorescence in situ hybridization, known as FISH, used to determine which chromosome are homologs, right? Because if you have what is a homologous chromosome? That means that they have the same genes and the same location or loci. It doesn't mean they have the same alleles, but they do have the same genes and the same loci, and that's what makes them homologous. So what does fluorescence on more than one chromosome indicate? Well, it can mean that you're dealing with homologous chromosomes, or maybe that there are multiple copies of this gene. So FISH is utilized to as a diagnostic tool to analyze genetic disorders, and can also be used to determine which cells in a particular organ or tissue are expressing a particular mRNA that you're looking for. So let's go and see how this looks. This is a very, very fancy karyotype where they have utilized a bunch of different probes. And so the different colors are corresponding to different probes. And you can see that the homologous chromosome have the same coloration, very similar coloration, and very similar banding pattern, which lets us know they have the same genes in the same location because they're homologs. Now over here, this is something interesting. This, for example, is maybe a tissue biopsy, and this is a cross section of tissue, and you can see that we're looking at individual cells here. And you can utilize FISH to determine which cells are producing or are transcribing a gene of interest, right? So for example, if we're talking about the pancreas and we're doing a study on cells within pancreatic tissue that are producing insulin, it's not going to be all of the cells of the pancreas. There are certain cells within the pancreas that are going to produce insulin. And so you can then go and design a probe to target that mRNA transcript that corresponds with insulin. Okay, the mRNA that will get translated into insulin, and so you can determine that. And over here, uh, this is kind of maybe what it would look like, where you would see some cells are showing, high levels of fluorescence and others not so much. So that's one example. But I know better. And what do I mean by I know better? Because this slide isn't that. This slide isn't that at all. But you would see something similar. This slide instead is the researchers are using um, fish as a diagnostic tool to be able to determine if an individual has chronic myelogenous or myeloid leukemia. Okay, so this is something that's interesting. When we talk about leukemia, there's not just one kind of leukemia. There are many, many, many different kinds of leukemia. And these different kinds of leukemia, depending on the mutation in these cancerous cells, they are treated differently. So leukemias are not all treated the same. They're treated differently, especially if you're hoping to get a good outcome with regards to your treatment. You've got to know what kind of leukemia does the patient have. And so what they're doing here is they're testing for chronic myelogenous leukemia. And what that has to do is with the translocation from chromosome number 9 to chromosome number 22. And so what are we looking at here? Well, what they do is chromosome number 9, the part that gets translocated, they're going to design a probe that will give off a red fluorescence. And for chromosome number 22, um, where the translocation would occur, what they're going to do is they're going to design another probe for that particular DNA, and it will give off a green fluorescence. And so, ideally, if there is no translocation, you would expect that the green and the red will be far apart. They will not be anywhere near each other because, there's no reason for chromosome number 9 to be right up against chromosome number 22 unless you've switched the DNA in a translocation event, which is what happens in chronic myelogenous leukemia. So if we look at this cell right over here, if you follow my arrow, if you look at this cell, you have two greens, which are separate from the two reds. That's what we would expect, okay? But over here in this cell, you got something that you don't want to see. You have the green and the red right next to each other abutting. And what that means is that there was a translocation here. And this over here is a cancer cell, okay? We can also see it right over here in this cell, okay, and there you go. So this, and if you're able to use this as a diagnostic tool before this cancer, happens to go and you know start to spread uncontrollably, uh, then that's a good thing. The diagnostics are very important because the sooner that you can detect a cancer, the better your prognosis will be. uh, the longer it takes to diagnose, the more difficult the treatment's going to be. Okay. And then over here, what this shows is, just once again, that what they're doing, chromosome number 9 is going to be associated with a red fluorescent probe. And that's the part that would translocate the chromosome number 22. Chromosome number 22, the part that would translocate, is going to be hooked up with a probe that will fluoresce green. So this shows a normal cell, where this right over here shows translocation, where the red and the green are right up next to each other, along with this, and this right over here, and this. And there you go. We'll learn more about this particular example when we do medical biotechnology. Okay, but for right now, we're just understanding, how fish works, fluorescence in situ hybridization. Okay, so with that, we are done for this aspect of our lecture, and now it is time to go and move on. So what we're going to start to do is learn about proteins. Proteins are a huge part of biotechnology, a huge part. I mean, the whole reason why we're manipulating DNA and learning about genetic engineering and learning how to clone and use restriction enzymes and get our transgene into plasmid and then transform, cells with our plasmid that has our transgene of interest is to produce a particular protein. It could be a therapeutic protein that can help patients with a particular disease. It can be proteins that are used in industry, whether it's the food industry, textile industry, uh, whether it's detergents. Uh, so proteins are huge. So that's what we're going to start talking about now. So let's go and let me share my screen and let's continue. Okay. Okay, so over here we got a question. We're going to start out with a couple questions. It says, what laboratory technique is used to study mRNA levels when a sample, mRNA levels in a sample when the level of detection is below that of northern blot? So you initially tried to do northern blot, but you're not getting, you know, you can't visualize anything on the gel. And that's probably just because your mRNA levels are too low. So if you can't visualize with northern blot, what might you want to utilize? And so I want you to put your answer choice in the chat. Is it A, southern blot, B, western blot, C, reverse transcription PCR, D, fluorescence in situ hybridization, or E, none of the above? So I'm going to give you about 10 seconds in order to do that, and we're going to go and move on. So don't forget, whenever a slide comes up, take your phone, take a picture, so that way, if you can't do all the notes that you really want to do, you can then go back at a later point and finish up those notes. Okay, let me go and stop and see what you guys are putting. Okay, looks like everybody, most people are putting C. I see a D, I see an E, but most people are putting C. So let's go back over here and take a look. Okay, what laboratory technique is used to study mRNA levels in a sample when the level of detection is below that of northern blot? Reverse transcription PCR. So what that involves is taking the mRNA that you do have from your biopsy, um, doing reverse transcription PCR to make cDNA out of whatever mRNA you have, and then performing PCR utilizing a forward and reverse primer for your gene of interest. And then, you can look at the bands on the gel. Once you run that out on a gel, the larger the band corresponds to the larger amount of starting material, the larger amount of cDNA you had, which directly corresponds to how much mRNA that you started out with. Okay, so let's go and move on to the next slide. This one says, what laboratory technique is used to identify which chromosome contains a gene of interest when generating a karyotype? Okay, what laboratory technique is used to identify which chromosome contains a gene of interest when generating a karyotype? And you have answer choices A, B, C, D, or E. So I want you guys to go and put that in chat. Okay, so you should be able to look at this pretty fast and figure it out. So I'm going to stop sharing and see what you guys are putting in chat. Okay, now it looks like a lot of people are choosing D. So let's go back over here and see what we got. Yeah, fluorescence in situ hybridization. That's what we just talked about. So if you wanted to use a laboratory technique to identify which chromosome contains a gene of interest, you would then design a probe for that gene of interest, and you would do fluorescence in situ hybridization with chromosomes laid out on a glass slide. Okay, let's move on to the next slide. And here's our last question. It says, what laboratory technique utilizes a probe with a 5' end reporter and a 3' end quencher to study gene expression? What laboratory technique is that? And once again, A, B, C, D, and E are your answer choices. So I'm going to give you a couple seconds to go and type that in. Okay, let's see what everybody's putting. Okay, now Looks like everybody's putting B. B is in boy. Okay, so let me go back over here. So this right over here is a TACMAN probe. And yeah, that's real-time PCR. So B is the correct answer. So let's go and move on. So now what we're going to do is we're going to start talking about proteins because that is, that's really what biotechnology is all about. Okay. Uh, a challenge of biotechnology is to understand and control the protein folding and the manufacturing of proteins. So, a big part of biotech is manufacturing proteins, and to do it at batch level, which is like massive, massive amounts of protein that can be used to help, people that have a particular illness. Um, or for industrial purposes. So we know proteins are complex molecules and they are built of chains of amino acids. And proteins have a particular specific molecular weight, and different amino acids have different characteristics. There are some amino acids that are hydrophobic, some that are hydrophilic, some that are negatively charged, some that are positively charged, and some that are polar and some that are non-polar. So amino acids have all these different characteristics. And that's going to determine the order of these amino acids and where these charged versus uncharged, polar versus nonpolar, hydrophilic versus hydrophobic amino acids are, where they are in that chain, that amino acid chain, will determine the conformation or structure of that protein with regards to how it folds. So structure and function of a protein depends on how that protein folds. If a protein is folded incorrectly, there's a strong chance that it's not going to be functional, and that could be a problem. And so if there's a mutation in a gene, a protein encoding gene, and that mutation leads to a change in a single amino acid, it can have devastating effects. Like when we talk about, for example, sickle cell trait, it's dealing with a mutation in the hemoglobin gene for for a particular substitution. So one nucleotide is switched out for another, and as a result, you get one different amino acid. And that one different amino acid and that whole big long chain of amino acids is enough to have a structure that is not an ideal structure and can result in sickle cell trait or sickle cell disease. Okay. Other proteins that, when folded incorrectly, that are associated with disease would be, for example, Alzheimer's. It's thought that some of the proteins, like the amyloid plaques, might be because of incorrect folding. Cystic fibrosis, mad cow with prions, improper folding, different forms of cancer, and even some heart attacks could be because of incorrectly folded proteins. So if we want to go and start learning about proteins, well, there are two big players, Pauling and Corey in 1951, that described two regular structures that are associated with proteins. And these structures are alpha helices and beta sheets. These structures are very fragile because how these structures form is by hydrogen bonding. And hydrogen bonding is a very weak bond. And if it's heated up, then those bonds are broken. So what we can do now is talk about the structural arrangement of proteins with regards to four levels. Now, when we talk about the alpha helix and the beta sheet, these are considered secondary structure. Okay, so what is secondary structure? It occurs when chains of amino acids fold or twist at a specific point, forming new shapes due to the formation of hydrogen bonds between hydrophobic amino acids. Okay, so whenever we talk about our alpha helix and our beta sheets, that's secondary structure. So then you might be saying, well, what the heck is primary structure? Primary structure is the actual sequence of amino acids in a chain of amino acids. So the actual, proteins are made out of amino acids. And so the actual order of those amino acids and which amino acid is where in that long chain of amino acids is going to determine, the structure of that protein. So there you go. Primary structure is the actual amino acid sequence. Secondary structure are going to be these beta sheets and alpha helixes that occur by way of hydrogen bonding. Okay, so let's learn a little bit more about alpha helix and beta sheet. These are very common secondary structural shapes. Alpha helix, amino acids form a right-handed spiral as hydrogen bonds are going to stabilize the structure by linking an amino acid's nitrogen atom to oxygen atom because of the hydrogen bonding that takes place. On the other hand, beta sheets, these are hydrogen bonds that are going to also be linking nitrogen and oxygen atoms, forming these parallel sheets. And sometimes these sheets are going to run in the same direction, and that would make them parallel. And when they run in alternate direction, they're going to be called anti-parallel. So over here, we can see the alpha helix, which is going to be a right-handed spiral. And we can see the hydrogen bonding taking place between the oxygen and nitrogen atom. And there we go. And over here, we see our beta-pleated sheets. Okay, with once again, hydrogen bonding taking place between the oxygen and nitrogen atom with the presence of, uh, of hydrogen. Okay, so now what we're going to do is take a look at the tertiary structure. Tertiary structure is a three-dimensional structure associated with polypeptides that are formed when secondary structures are cross-linked. And you might wonder, how are they cross-linked? I mean, how does that work? Well, how do you cross-link these secondary structures to get this tertiary three-dimensional structure? You are going to do this by, for example, disulfide bonds between cysteine residues, uh, and these bonds are strong and they're not easily broken, and so that's our tertiary structure. So tertiary structure of a protein determines its function. Uh, whenever we talk about a protein, like for example, if it's an enzyme, enzymes have an active site, and if the protein doesn't fold correctly, that active site is not going to be able to hook up with the proper substrate that the enzyme needs to work on. So that tertiary structure is very important. It's what determines the function of the protein. Okay. But now there's something else that is called quaternary structure. And quaternary is when you have uh, more than one tertiary structure. And how does that happen? Because you actually have more than one protein coming together to form a tertiary structure. For example, when we take a look at hemoglobin, if you've ever looked at a molecule of hemoglobin, it is actually composed of four different strings of amino acids. And each string of amino acid is going to have its own tertiary structure, but then those structures come together to form a quaternary structure. So quaternary structure is a unique globular three-dimensional complex built of several polypeptide chains, several chains of amino acids. And why are proteins so fragile? Because of, the hydrogen bonding, because they can be easily broke. So over here, this is in our textbook, and it shows the different levels of organization with regards to protein structure. And so over here, we have our primary structure, which is just the order of amino acids within a chain of amino acids. We have our secondary structure, whether it's alpha helix or beta sheet. When the secondary structures come together by way of disulfide bonding between cysteine residues, you would have your tertiary protein structure. And when you have more than one tertiary protein structure coming together, then you would have your quaternary protein structure. Okay, so this right over here is very helpful. This is known as a ribbon diagram, also known as a Richardson diagram. And the colors and the shapes give us information. So for example, the areas that we see in red, where you have this right-handed spiral, these are your alpha helices. okay? And the areas where you have green, these are your beta strands, your beta pleated sheets, okay? And then you have other things associated with the protein, like for example, you have what's known as post-translational uh, modifications, and that may include the adding of sugars or other molecules that are important with regards to the function of the protein. Okay, like for example, hemoglobin, right? You have that iron-containing heme group. Okay, and so that is a structure that comes into play uh, after that translation occurs. So an example of some of these post-translational modifications would be glycosylation. So glycosylation is the adding of carbohydrate or sugar units to specific areas on a protein. And that can affect, number one, um, the longevity of the protein. How long is that protein going to be around before it gets recycled or eliminated? It can also be a factor in determining the solubility of the protein and also whether or not that protein is to be oriented into or out of plasma membrane, for example. And there are over 100 post translational modifications that occur within eukaryotic cells. Okay. Now, it turns out that prokaryotic cells are capable of post translational modifications, but not necessarily the same exact post translational modifications that eukaryotic cells are capable of utilizing. So over here, this is another example of a ribbon diagram or Richardson diagram. And you can see that over here, it looks like we got a little bit of alpha helix. We got some beta sheets over here. But then we have these glycosylating sugars, which is a post-translational modification to make this protein functional. So an example, why do we care about, post-translational modifications and whether or not you have a protein that gets glycosylated? Well, we can think outside the box, and when we have information, we can then co-opt and use that information for beneficial purposes, like for treating disease. So for example, glycoproteins can be used as a new way to target and destroy B lymphoma cancer cells. So in the past, maybe like 30, 40 years ago, when somebody had lymphoma, it was pretty much treated the same way. You'd have these very, very harsh chemotherapies, and it would target all rapidly dividing cells, whether it was a B lymphoma cancer cell or it was hair cells or other cells in the body. And so the whole idea is basically this treatment would uh, kill a whole bunch of cells in the body, and the whole idea is to have it do as much damage as possible to the B cancer cells without dying in the process. Okay, but that we've learned now that we have much better diagnostics and different techniques to target cancer, that's not the best way to go. And when we talk about breast cancer or leukemias or lymphomas, there's not just one kind of breast cancer. There's not just one kind of leukemia or lymphoma. There are many different kinds, and it's important to diagnose which type of cancer you have so it can be treated most effectively. So, for example, how does this treatment work where we're going to utilize glycoproteins? So, it turns out, for example, on these B lymphoma cancer cells, they're going to overexpress CD22, which is a co-receptor that these B cells are going to display. Okay, the CD stands for clusters of differentiation, and it's CD22. So if we know that B lymphoma cancer cells overexpress this particular receptor that's on the outer surface of B cells, and we know that this CD22, that this receptor targets particular glycoproteins, then with that information, we can be very clever and use this for our benefit to target those particular cells. So what we can do is we can figure out what glycoprotein hooks up with CD22, and we can combine that glycoprotein with a nanoparticle that is loaded with a particular chemotherapy drug. And so this glycoprotein with our nanoparticle is going to be specifically targeting those B lymphoma cancer cells, leaving other cells alone. And that is something that is really good. So by targeting these particular cancer cells with receptors for these glycoproteins that are attached to nanoparticles that are loaded with a particular chemotherapeutic agent, the effective dose of the drug can be increased while protecting normal tissue. Okay? And that is something that is amazing. Is targeted chemotherapy. It says, why might normal chemotherapy not be as effective? Because normal chemotherapy targets all rapidly dividing cells, and it does a number on the body. It devastates the body, and if we can target just the cancer cells that need to get eliminated, that is by far the best way to go. So over here, what we see, this is like a for fun slide right here. And PTMS post translational modifications, we said there's over 100. These are just some of the ones that you're most frequently, uh, you know, that you would most likely come across maybe in a paper or maybe, in research. So phosphorylation, adding a phosphate group. Acetylation, remember acetylation, how we can acetylate histone proteins in order to uncoil the DNA in the area. There you go. Glycosylation, we just learned about. Nitration, adding a nitrate group. Hydroxylation, adding an OH group. Methylation, adding a methyl group. So these are just some of very common post-translational modifications. Okay, so now let's go start talking a little bit more about protein production with biotechnology. Okay, now it says use of proteins in manufacturing is a time-tested technology. Yeah, if you like alcoholic beverages, how do they do that? Well, they utilize certain enzymes and proteins. So that's heavily incorporated in, alcoholic preparations, whether you're, doing brew, beer brewing or winemaking. Uh, for example, cheese making. How many people like cheese? I'm going to stop sharing right now. How many people like cheese? Raise your hand. Okay. A lot of people like cheese. Maybe cheese doesn't like you or your gastrointestinal tract, but we like cheese. Okay. So now, okay, put your hands down because I'm going to ask a different question. Put your little cartoon hands down. Okay. What would you do if I told you that cheese, in order to go and perform the cheese process, where you take milk and you denude the casein proteins, which are milk proteins. You're removing the carbohydrate, you're splitting the casein proteins so they can coagulate. And why do you want it to coagulate? Because that's how you're producing the curds, which is what you need to make the cheese. The whey is like the waste. We don't want the whey, we want the curds that are going to be used to make cheese. What would you do if I told you that where do you get the enzymes for that you get in the past? We got enzymes to make cheese from baby calves and goats. So we would take baby calves and goats, when they're babies, are super cute. A big old cow, I don't know how cute that is. A big old goat, I don't know how cute. But the babies are super cute. Like if I saw one right here, I would want to go and hug it because they're so little, they're so cute, they're adorable. What would you still like cheese if you knew that in order to make cheese, you would have to kill the baby goat and baby cow just so you can do a protein extraction of their stomach in order to get a particular enzyme that is necessary for the cheese making process? Who would still like cheese? Who would still go out and buy cheese? Okay, so Leanna, and Kenneth, okay, they really like cheese. They're like, forget that, man. Man, I live off of pizza. So I'm sorry, those little baby goats and baby cows, we're just gonna have to go and slaughter them in order to get the enzyme. Okay, and the enzyme uh, is basically called chymosin. Okay, but what would you do if I told you now that every time you have pizza, no worries? Because guess what? That's not where we get the enzyme anymore. What we were able to do is to go and take that gene, take that DNA, and get that into a plasmid and transform bacteria, then can produce batch amounts of chymosin, more than we can ever need. So we never have to go and kill a baby goat or baby cow in order to get that enzyme from their bodies. And why do they have the enzyme? Because baby cows and baby goats need that enzyme to be able to go and get the nutrients from their mother's milk. Okay? So how many people are happy now that we can do biotechnology and we can get as much enzyme, as much chymosin enzyme as we want without killing baby goats or cows? There you go. So next time you have your pizza, nothing that you have to worry about. What I want to do right now, because we are running out of time, I want you to go in your textbook and look at this particular figure. And what this figure shows is how they actually did this. How they actually did this. The step by step protocol as to how they can use bacteria and grow bacteria to produce our transgene of interest. And what is our transgene? It's going to be a gene that codes for the enzyme chymosin that will denude casein protein from glycopeptides
Hello, you're
and
Brian Peters.
split that casein
Yes, hi, how are you
protein
doing today?
so we can go and get the curds needed to go and make cheese. And cheese isn't, besides alcoholic beverages and cheese, we use recombinant DNA technology to produce a whole variety of enzymes, hormones, okay, whether it's growth hormone, whether it's insulin, you name it. We use biotech for these purposes, and even antibodies like monoclonal antibodies we can generate utilizing biotechnology. Okay, guys, we are out of time. We are out of time. So this is where we're going to go and stop. I will be around to ask questions. I will have many office hours right now. And so I'm going to stop right there. And if